Hello, everybody. Today is November the 20th, 2020, the year that continues on giving. We have some great news this week on the vaccine and also some information on new treatments. And unfortunately, we have some horrific news on how the virus is spreading in the US, and Europe is also bending the curve. Just as a reminder, it is my background is described on the trailer, the description of every video, and also in the About tab. I am not a doctor. I've been working in healthcare for 40 years, more on the business side. My objective is to be a reader digest. I don't make the news, I don't make the facts, I just report the facts in a way that hopefully is digestible and that you can be more educated and make informed decisions. So please stay healthy and enjoy Thanksgiving, and let's get the show going. Hello, this is the second video of November the 20th. I'm going to be focusing on the clinical update. We have a lot of great news to talk about. Vaccines are working. 2 of them have now reported amazing results. Some new data on antibodies seems that they're lasting longer, so that's also great news. There's some new data showing that smokers may be at higher risk. I'm going to also have a lot of update on the diagnostics and on epidemiology. I know it's a little bit depressing, but this is the reality. There's a lot of interesting tools that you can use to see what's your chance of exposure, and the links are going to be posted on YouTube. And also we talk a lot about collateral damage of COVID when people are dealing in care, and how that's impacting people being at risk of cancer and burnout and other issues there. So let's look at some of the better news. So just as a summary, we all are going to be exposed to COVID. I think that's a very high probability. The key thing is not avoiding exposure, is avoiding being infected. And I think I really want to, again, spend a little bit more time, and then you can look at the video 2 weeks ago. What are the areas that are low risk? More and more data are showing that contaminated surfaces, except for high traffic doorknob and elevators, are really low risk. And we'll talk more about that. Outdoors, if you're out, with a mask over 6 feet, there's a lot of dilution in the open air. So very safe environment there. What you need to really work on is to minimize the amount of viral load you get exposed. And that's a combination of how much you get from that person, what's the amount of time, and now they're talking about 15 minutes cumulative during the day. And what's the environment around you? Is this poor ventilation, crowded area there? Are you close up to the person, less than 6 feet? And are you in a super spreading event, in which we'll talk about that. And then what can you do to decrease the amount of viral load you get? Because we could all get some virus, but if you get to a certain amount, that's when you get infected. So you want to decrease the amount. And the mask is not only to protect other people if you're asymptomatic, but it's also to protect you from getting viral load that's in the air. Distance is critical, and ventilation. Be aware of your environment. We discussed this in the video today on the epidemiology. There is that the prevalence of where you live by county is critical to keep an eye on, because that will affect how many people you can see that safely there. Ventilation, essential. Spend some time in opening your ventilation, getting some air filter, HEPA filters, and really work on ventilation there. Be aware of your ventilation when you go to a restaurant or if you go to somebody else's house. Be aware of your environment. So I'm not going to spend too much time. We spent quite a bit of time in the video on November the 6th. Please go on YouTube and check it out. Okay, so this is Hong Kong. They developed little robots there to basically disinfect as a shopping mall. And what I think we're learning more and more, if you remember in March, we were all freaking out about, is it on the metal? Is it on the plastics? Can we even open the box from Amazon? Should we clean all our food from the Safeway, from the supermarkets? And I think more and more the CDC is coming out that, yes, there's a very low risk, but really the biggest reason why people get infected is the airborne droplets. So you don't want to be overboard, in using Lysol for everything. And I think the data is coming out from Hong Kong and other places that a lot of people are using antimicrobial coating and had a fleet of robots and clean surface. But the perception is that there's been, it doesn't really have a huge impact on mitigated virus spread. What's really important is ventilation and indoor filters, because this airborne droplet stays in the air for hours. That is really where you have your highest risk of infection. So another data just came out about something we've discussed multiple times, is that 85% of the infection are coming from 10% of super spreading events. And there's a modeling study that was just been published in Nature, and where they did analysis on 98 million people in 10 metro area there. And what they show, for example, in Chicago, which I picked some of the graphic here, is that the area that you're really at risk is religious organization like a church and the services, indoor, full service restaurants, so indoor restaurants, hotel and motels are all higher risk there. A little bit on the hardware store and grocery store, so still be careful depending on how crowded these places are there. So these are the area that are really the big drivers in the transmission rate. And so the takeaway of the studies to says we don't want lockdown. We just want to be much more precise in the intervention. So as you see, people are starting to shut down the bars after 10 o'clock or the indoor restaurants after a certain time there. And that may be just much more effective than locking everybody up in their home. Another interesting study showed that humidity and heat is your friend. So if you are in Arizona, like some of my friends here, and if your temperature over 75 degrees Fahrenheit and you have over 50% humidity, it basically, the virus doesn't last very long. And just because the particles in the air, basically disappear and they drop very quickly there. The virus loves cold temperature. That's why we're having that winter, wave here. And again, one of the things that people have been experimenting with, is to have higher humidity inside the house or in the environment there, because when you have high humidity, the droplet gets bigger and they drop faster. They don't stay in the air as long. And if you remember, we discussed this, it's one of the prior videos, how long it stays in the air. So new data just came out on smoking. This is a little bit confusing. If you remember the last few months, there was exactly the reverse data being published, which is if people were in hospitals there in New York, most of the people who were hospitalized had a lower incidence if they were smokers. So there was a perception that, hey, maybe nicotine is protecting you. If you remember, we talked about this. And this data came out from UCLA, showing in much more detail. Before, the prior data were just reporting outcome as opposed to really understanding what's called the mechanism of action. And what they have shown is that somehow the smoking is interfering with what's called interferon. Interferon is kind of the key thing we're going to talk more and more about on how to basically boost your immune system to fight the virus. And so what they did is that they did an analysis, and there's another study from Stanford showing that just came out around the same time, showing that if you're a smoker, you have a 5 times higher risk of catching COVID. And it's 5 times higher risk if you have e-cigarette, and if you smoke normal cigarette and vaping, it's 7 times risk. And this is to give you an idea of what's going on. In green is, unfortunately, our little COVID, and then in blue is the airway, is the cell nuclei that you have in the lungs. And what they're showing is that in this next graphic here is that when you're smoking there, you somehow slow down the production of interferon in your lungs. And when COVID comes, it basically has an easier pathway. And then they did an experiment where the person was smoking, and you can see if you're smoking and you get exposed to COVID, the damn thing is everywhere in their cells that they identify. But if you give the person interferon, it goes away. So they confirm it 2 ways. One is to show there was a lower amount of interferon. Number 2, as a treatment, if they give interferon, they can see in the Petri dish, in this case, I think, that they could get rid of the COVID. So the belief is that people who are smokers, even if they're young, could be at significantly higher risk of catching COVID for the same amount of exposure that somebody else may not catch COVID. Because remember, we are going to be having viruses going to our lungs. The question is, can you stop it before it basically multiplies? Or basically you don't have a response from interferon, and then it multiplies, and then you have a full-blown infection. Let's talk about vaccine. And again, I did a full review of all the vaccine on September 11th, so please go check that video. Let's do an update. Some great news. Moderna has just updated their phase 3 initial results, showing 94.5% effectiveness. That is absolutely stunning. Honestly, people were hoping to get 50%, and it's a new technology called mRNA, which allow 2 companies, Pfizer and Moderna, to develop the vaccine extremely quickly in the fastest time ever, I think, in the history of making vaccine. The way they did the calculation there is that they had 30,000 patients. Half of them were on a placebo, i.e. it's a control group, and the other half got the vaccine. Of the control group, 90 people got infected, and 11 were severe, and the people who got the vaccine, only 5. That's how they do this ratio, and they end up with 94.5%, kind of simple math. They are planning to do EUA submissions there. And I think, just to explain a little bit what the mRNA is, it's a new mechanism of action. It's not the traditional way of how we do a vaccine. We typically trick the cells into producing a bit of a protein that looks like the piece of the virus. So it's not like a live virus. It basically synthesizes, and we fool the body. As a result of that, the immune system kicks in. It recognizes what looks like an invader, and it builds antibodies. And when it builds the antibodies, then when you get exposed, let's say, a month later there, now you have a defense system to the real virus there. It requires 2 injections at 21 to 28 days apart. And the good news about the Moderna, which is different than Pfizer, is that it can be stored at normal temperature of minus 20 degrees C. Because Pfizer needs these super, super, super freezers, which most people don't have. And so that means most doctor offices have that capability of freezing them at the temperature there. It also can be kept in a refrigerator for 30 days, where Pfizer can only be kept for 5 days. So we should expect Moderna to submit the EUA literally in the coming days, and probably be approved, sometime in December. Another great news is Pfizer, shortly after Moderna, came out with 95% effectiveness. Again, amazing numbers, similar technology. The key thing is that they reached the 2-month safety on 43,000 volunteers. They had 170 COVID cases, 162 in the control group and 8 in the vaccine group. So astonishing big differences in the number. It's interesting, they had 10 severe cases. And in this case, they had some severe cases in the control. 90% of the severe cases were in the control group, so very similar to the data we saw in Moderna. And this is great because in their case, they had a similar effectiveness for people of the age of 65 years old. One of the worries that, as you get older, your immune system is not as strong. And there was a concern that some of these vaccines may not provide enough antibodies for some of the older population, which is the one at higher risk. And the good news is that there's a very strong antibody response. And also very nice, there's a very widespread distribution racially, and also using different age groups though. So it's really great news. They filed literally today on November 20th for the EUA. They are planning to do 1.3 billion doses. Remember, you need 2 doses per person there. It has to be in this very low temperature there. So they've built some special thermal shippers using dry ice to keep it at this really low temperature there, so that if you continue to use dry ice, you can keep it for 15 days. And it has a GPS-enabled thermal sensor to track the location temperature, so that if you do get it, you know that during the transits and the transports, has been safe, because otherwise you don't know. So what's the process? The FDA has called a meeting on December 8th to the 10th, so it's going to be 3 days. They expect to probably, I guess, reviewing both companies, but at least officially Pfizer. After they make the announcement, which we're assuming will come shortly after that, the earliest will be the 10th, they'll be immediately released for distribution in the US. And there's already been some stockpile in the US. And then there's a group called the ACIP, and that's within the CDC. And it's the Advisory Committee on Immunization Practice. And they are the one that basically set the rules on the rollout and who gets access. They are planning to get together within 24 to 48 hours after the EUA is announced, so mid-December. They will decide the priority and the exclusion rollout. So some people, for whatever reason, based on the data that was submitted to the FDA, may not be eligible. And then within 24 hours, everything has been set up with the task force to start shipping to hospitals. So what do we know so far? There was a recommendation in the summer that came from the CDC. This was their expectation. Again, they could change their mind when they review this in December. Phase 1 is going to be healthcare workers, and we need them because we have this massive outbreak. So we need to make sure we have as many healthcare workers. So they'll be the first one. And that's around 12 million people. So that's 25 million doses. And then we need to also protect the elderly, especially people in nursing home with this, overcrowded facility there. It's been, if you remember, 41% of the US fatalities are people in nursing home. And then people who have underlying condition, high risk. So these are people probably immunocompromised, or people maybe have had cancer treatments. That's the first group. And so then we have the phase 2, the teachers and the school staff, phase 3, young adult and children. What they're working is breaking down these big 3 buckets there into smaller sub buckets. And for example, in the UK, they literally are going to be releasing by age group. So 85 end up first, and then 75 to 85, and then 65 to 85. I don't know if the US is going to do the same thing. But you should expect that this 110 million, which is the first batches there, which requires 220 million doses there, is the one that's going to be in a rollout there, and before it goes to the general population. There was an interview from CVS today, a CEO, that basically said he expects CVS and all their stores is under contract to basically give it to the general population, and they expect to start probably doing this in the March timeframe, just to get the expectation for the general population there. Now, let's talk about the Chinese. If you remember, Chinese have two companies. One is called Sinovac. An article in Lancet, which is a major journal, just got published this week showing their phase 1 and 2. Phase 1 and 2 trials are not for efficacy, but for safety and maybe early signal of efficacy there. And what's interesting is that they show lower antibody level than what we have seen in survivors or what we have seen in Pfizer and Moderna. Pfizer and Moderna have slightly higher antibodies than in survivors. And so it may be effective, but the amount of the duration of protection may not be as long. Remember, that's the one thing we don't know from Moderna and Pfizer is how long the antibodies are lasting, because we only have data for 2 months. In addition to that, the Brazil equivalent of the FDA has suspended a phase 3 trial recently, a week ago, due to some serious adverse event. Now, that happens a lot. It may be unrelated. We just don't know. So both Sinovac and Sinopharm, the two leading companies there, are using inactivated virus. It's a different technology. It's a more traditional way of the way you do a vaccine. So you grow a virus in a petri dish, and then you weaken it. So it's the COVID virus there, and then you basically you can store in a refrigerator at low temperature there, which is kind of easy to do that. And then Sinopharm, which is controlled by the government there, has vaccinated 1 million Chinese citizens. And what's really surprising about this is that they have not released any efficacy evidence. So maybe that's their phase 3. Because they have a low incidence in China, most of their phase 3 trials that are in process right now are done in countries outside the US, in South America, the EUA, the Philippines. In China there's a market there, which is a consumer market there, where you could buy this vaccine for $30 per shot. So roughly you need 2 doses, so it's $60. So the Chinese population is buying it. That's how they're getting to these 1 million citizens there. One of the big things they did is that there's a lot of Chinese workers going in Africa to work some of the mineral mines. And all of those Chinese workers who are leaving China are all being vaccinated by the government. So how long is the immunity? Now, a lot of us, remember the whole idea that you get measles, the mumps, and rubella there. It's pretty much for a lifetime. This is not the case for COVID-19. We don't fully know yet. What we know is for SARS in 2003, the immunity was around 36 months. We know for MERS, which was a different form of the COVID, that was 34 months. So there is a hope that, we may at least have 1 year, but, we still need the data there. But it's going to be a short-term immunity. It's not going to be a lifetime protection. That is a sure thing, at least as sure as we know by the science there. So we also know from hepatitis viruses that antibodies are not always protective. Some people have antibodies, but they may get reinfected. Some people have a very fast drop in antibodies and may be at higher risk than others there. So some good news there. So if you remember, the IgM is the first antibody you get in the first few days after you get exposed to the virus. And then over 21 days, you start getting what's called the IgG. The IgG is the antibody that peaks at 21 days, and then that's supposed to be the one that protects you for months. That's the one they've been measuring there. And what they have shown is that in this research from La Jolla Institute, they look at 185 survivors that give blood samples, is that there has been a slow decline. There is a decline, but most people still had some antibody at 6 to 8 months. What is a bit worrisome is that you had a differential from person to person of 200x. So it does mean that if you have been exposed, you should probably get yourself tested for antibodies, within maybe 3 to 6 months, just to see what is your level and what your decline is. Now, there's the spike IgG is stable at 6 months, slightly higher for men than women, but that could just be the sample there. But the memory B cell, which is the one that we can really have a long-term protection there, has been going up. At 6 months, it was higher than 1 month. Remember, some of these are kind of going up as the body is building its immune system there. And the T cells, which is the famous T cells, which is less specific than the antibodies there, they have a slower decay with a half-life of 3 to 5 months. That means you have lost 50% of your antibodies at 3 to 5 months. So clearly it's showing that this is not a lifetime protection there, but maybe 6 to 12 months. This is the thing to keep an eye on, that 10% of these people there had no long-lasting immunity. So, I think we're going to discover in the testing that as people are getting exposed to it, they need to be checked afterwards. So autoantibodies is a bit of a concern. We have done an analysis that was published in Science where they showed that 10% of patients having severe COVID, so these are people who are in hospitals on oxygen, have an antibody that has disabled the immune system and basically shut down that interferon I mentioned earlier. Interferon is really kind of the key fuel for defending your system. And it turns out it's a silent condition. These people had no idea they had that until they got infected. And they start doing an analysis backward of identify why are some people getting sicker than others. And they discovered that 3.5% of the severe COVID had that mutation that basically stopped or had an impact on the amount of interferon that's being produced when you get infected. And it's really important there because interferon is like a fire alarm. It's like your sprinkler when you have a fire in your house. It's the first thing that the body responds to when it gets exposed. And that's the most important part because that's when the virus is proliferating and exponentially growing there. That's when you want to stop it there. There is worries there that these autoantibodies, are kind of part of autoimmune diseases. And so, for example, we know that people with type 1 diabetes and rheumatoid arthritis, are at higher risk of autoantibodies there. This is the interesting part. 94% of these autoantibodies are in men. I repeat that. 94% of these people were men. And there's a perception there that it's tied to the X chromosome. And as you know, the men have only one X chromosome, the women have two. So even if you have one chromosome that's not really working well on the interferon, you still have another one that's a backup. And that explains why we saw in the early days in the spring a higher mortality rate on the men. So this is a bit of a graphic there showing that when you see the SARS, you have this high percent of the population there that they have normal interferon production and all of that. And then suddenly you have these people there that not with demographic, but suddenly have these life-threatening severe cases. And when they analyze them, they find out that 10% of those people there had that basically mutation that they did not know about. They had no problem before about it. It was just affecting the production of interferon due to that COVID. So let's look at testing. Just a quick reminder there. So, you get exposed over here and you have a period of incubation, which we know could be between, 3 to 7 days, depending on the cases there. This is unfortunately the period there where you're the most infectious. And this is when, and we talked about this 2 weeks ago, that the PCR may not always be accurate. It may take up to 4 to 5 days before your PCR positive is correct. So if you get a PCR test too early after you have been exposed, and let's assume you meet somebody and they tell you they're positive and you get tested the next day, that doesn't mean you're not infected. So again, there's a person there with PCR can't be trusted. This is this period there between 5, to maybe 15 to 21 days. Then afterwards, basically, the virus goes away and the PCR will be negative, but you're in the middle of fighting the condition of COVID. So this is when during that period there, you want to do the PCR, the swab CPR there, and you don't want to do it afterwards. If you get infected there, you start building this antibody. And remember, the IgM very quickly drop. And the IgG are the key one I just discussed about. That's the one that starts peaking at 21 days. So this is when you want to do the antibody detection. And I'm explaining this because there's 2 types of tests. There's the PCR that's basically looking for the virus piece, and that's only accurate during this window. And then there is the antibody, which if you do it too early, is going to be also negative. So you really want to really understand, and I know at times it's hard to do, when you have been exposed, and typically if you have symptoms, it's 3 to 5 days before you have your symptoms there. So understanding this curve is really important in knowing what type of test you want to do. So there was a very interesting presentation done by Quest Diagnostic there. And so I want to kind of refresh some keywords: sensitivity is the ability to detect the infection when it's present. If you have 90% sensitivity, that means that 10% of the time you will not detect an infection. That's called a false negative. If you're specific, it's the ability to detect only the infection. And so that means that at times you can have false positive results. So what we typically want to make sure is to minimize false positive and false negative. And you may hear this terminology, sensitivity and specificity. So that's how you basically tie the two together. So a false positive, it means a test indicated you're positive, but you don't have any infection, which does happen with the antigen testing. They have up to 30% of false positive. A false negative means that's even worse. You think you're fine. On reality, you're infectious and contagious. Okay. So this is prevalence, is what I discussed in the first video today, is what's the amount of case in the population there. If you have a high incidence and a high prevalence in your county there, you have to take extra caution there, like in North Dakota and South Dakota. Okay. So there are three types of tests. There is what's called nucleic acid amplification test. This is roughly the PCR that we all have been talking about. And this basically detects specific genetic sequence of COVID. Very, very, very specific. If it's positive, you're infected. Okay. Then there's the antibody. And the antibody is what we discussed, this IgG. And the IgM is detected soon after the infection, then quickly go down. So really the IgG is what you want, but remember, it can take up to 21 days before it's detectable. So if you have symptoms there, that's not the one you want tested. What you want is to find out if you're still contagious as you get discharged. And then there's the antigen, which is confusing everybody. A lot of the point of care, there's really fast 15 minutes turnaround antigen testing. And what they do is that it involves an antibody that is specific to a target antigen. That is basically usually the spike of the virus there, and it's basically recognizing them. And this is the problem we have been having there, is that these very fast tests there have a lot of false positive. I'll talk more about that. So Quest did this really good webinar there, and this is coming out of the CDC, that look at the difference between both sides, because I know it can be very confusing. So this is the PCR. This is the one you do the nasal swab, and a very high sensitivity and specificity, but it takes two to several days. And right now there's such a demand that we're again in backlog. I mean, I'm sure if you watch the news, people are staying for hours in line, trying to be tested there. So we're back to where we were in the spring there. The antigen testing are very fast, 15 to 20 minutes. But if you are positive, you should follow up with a PCR test just to make sure it's correct. And so the good news is that there are some new tests that have come on the market. So Quest has released a point of care rapid antigen testing. So it's being done by clinicians there. You can do a nasal swab. You can see they go way up there in the nose. And then they put into their machine, and they put into this device there, and it will, like the pregnancy test, it will tell you if you're positive or negative, depending on the lines that you have there. They claim 97% sensitivity and 90% specificity there. So the good news is that they're starting to release this test and distribute it to the providers. So it's going to be easier for the providers to do this type of testing there. And they're also working with drones in Las Vegas. I had to put something humoristic this week. And they're working with Walmart, and they're going to be doing, I think it's a 1 mile radius to do a pilot there to see if they can deliver some of this testing there to single family homes. So just for the entertainment of the week. Brand new news, 2 days ago, Lucira got approved for the first at-home test. It's a test there that is nasal. So you can self-administer the collection. It's only for people over the age of 14 years old. It's using a technological lamp, which is kind of similar to PCR. It amplifies a specific piece of the genetic material to detect it. The reason why it's so much nicer is because it only needs a single temperature, of much easier process there, so therefore you could do it at home. And that was the big breakthrough we've all been waiting for. You get the results in 30 minutes. You literally get a little light that comes up, that's being shipped with the kit. The kit is around $50. Initially, it's under FDA approval with prescription. So a physician has to write your prescription. The initial rollout is going to be done by Sutter Health in Northern California and the Cleveland Clinic, which were the two primary investigators. They expect to get a national rollout in the spring of 2021. And that's what we've been looking for, is a way for you to be tested at home. Right now, they will need to get additional FDA approval to be able to online ordering. So right now, it's only available if your physician or your healthcare system ships it to you. But the data is fantastic. Look at the positivity there. That's what we've been waiting for. And because of the traditional antigen testing like Abbott and Becton Dickinson, a third of the nursing home are not using the testing equipment that was shipped to them by the government. If you remember, the federal government spent over $100 million to basically ship all this testing equipment to nursing home because we have such a big outbreak there. And only 30% of these nursing home, 30% of these nursing home have not done a single test. And the reason is because it requires a lot of documentation. If anything is positive, you have to keep track of it, you have to send it to the government, and they're totally overwhelmed right now, and a lot of them are understaffed. And the bad news is that there's so many positivity that are false that you have to do a PCR, which is an extra step. So some states like Massachusetts have discouraged the use of this rapid antigen testing because it's totally overwhelmed the nursing home. So that's why it's great news that these other tests just got approved. So as a result of that, this is the layout. If you're doing what's called screening and surveillance, you're using this antigen test, if you have anything positive, you have to do a PCR again and repeat that test, just to make sure that you are indeed positive there. And of course, you go immediately into isolation there because it may take you 2 to 5 days to get the results of the test. So I want you to make sure that if you do the IgG test, and if it's positive, so if you do the antigen test and it's positive there, you should follow up with a PCR. And that's for diagnostic. And then there's surveillance, and surveillance is basically we test a large part of the population and we try to get what's called the prevalence, which is what's the percent of the population that is infected. So this is what's called serology. So this is the number of cases that have been officially reported. These are the ones that have been confirmed by PCR. And then it has been estimated could be 5 to 10x more people basically have been exposed to the virus. They were asymptomatic. They basically were not tested at the right time. And so right now, when you talk about serology, it's not diagnostic to make sure you should get treated. It's just to see what the incidence of the population is. So again, the IgG, you can see, is really peaking at 21 days. And this is the good news. You can see it's staying pretty constant after a certain period of time. So more and more data seems to be more positive. As time is passing, we're not going to have a feeling for what's happening to these antibodies. So let's talk about treatment there. A quick update of what's going on. I did a full review of all the treatment on October 23rd video. So please go take a look. There's a protocol done by EVMS, and it's a group of critical care doctors there who have been very active. It's Dr. Paul Marek in organizing a group of ICU doctors there in coming up with protocols. Let me quickly do a recap of what we talk about. You have that incubation period there. We have the viral replication there. This is when you're the most contagious there. And you can see by day 11, they are assuming in their cases there that it really, is pretty much no longer there. This is asymptomatic there. And this is when some people have a delay in innate immunity. This is when the interferon is so critical in stopping the proliferation of the virus. And that's when you get into the cytokine storm and you end up, unfortunately, with problem with breathing and maybe in a hospital there. But keep in mind that during that period there, you still have viral debris that are coming out. So you could still test positive with the PCR, even if you're not infectious. Because PCR doesn't test if you're contagious. They just test if you have viral debris. So typically, if you look at clinical symptoms, this is when you have the first symptoms there. Then this is when you're at risk of getting hypoxic. Anything below 94%, you have to get medical care ASAP. And this is when you end up maybe on the ICU on special oxygen there if hypoxia is getting worse. So this is when we give the antiviral drugs that we've talking about. This is when we give the anti-inflammatory to basically slow down the cytokine storm. And methylprednisone is really the one drug that so far is working the best there. And this is when giving interferon earlier, when you're basically having that first innate immunity response is so critical there. So a quick review of what's going on. Oxygen saturation there should be between 95 to 97% at sea level. It will go down if you're in elevation. And then when you're having that early phase, everybody should have a pulse oximeter at home. And as some of you know, I launched pulse oximetry, I hate to say 35 years ago. This is when you have to worry. If your oxygen saturation is dropping, your heart rate, your resting heart rate is going up, you may be at risk there. That's when you have to see a doctor and get yourself tested and even go to the ER. And when it drops like a rock there, this is when, unfortunately, it's really at high risk. And this is kind of correlating with inflammatory response. So they use this medication, the different type of medication, different time. So it's really important there that you see a doctor as soon as possible. So this is their protocol, which is, as people are deteriorating there, the first time with low flow oxygen, we've learned from New York that you want to avoid intubation if you can. Then you increase the flow of the oxygen there and you start giving more type of drugs for inflammation there. We do the proning, you may have heard, which is you put people on the stomach. Very often you anesthetize them or sedate them severely there just so they don't fight, being in that position there. And then unfortunately, if you continue to deteriorate, then you go into invasive mechanical ventilation there. And then you go into, everything in the kitchen sink. And so the World Health Organization today came out, or yesterday came out against using Remdesivir. Remdesivir is a drug that was produced by Gilead. And they published this in BMJ, which is a British journal. And they came to the conclusion after having looked at 7,000 patients being hospitalized with COVID there using experts that Remdesivir is a very expensive drug. It's intravenous and it doesn't really provide enough benefits to being recommended, and that they should use those expensive resources in better ways. Of course, that's literally a month after the FDA has approved Remdesivir under high political pressure, if you remember. And then they just announced yesterday a new EUA for combination therapy between Remdesivir and a type of rheumatoid arthritis drug. And I'm not going to try to say that, baricitinib or something like that. So again, I mean, not great news there. Some good news, interferon. Remember, we talk about interferon. Company called Synergen is making a form of interferon that could be used, and this initially was developed for multiple sclerosis. And they have shown early data, and it's basically, it's an inhaled formula, so it's an easier way to give it to the patient there. And at 48 patients versus 50 placebo. So it's not a huge trial, but it's significant enough. And this is the amazing thing. They decrease the odds of severity of death by 79%. And the only data we've seen is like methylprednisone is the one that's showing this type of huge impact there. It had no difference in the time to hospital discharge. So it's being used for people who are in the hospital setting. So they're already there. And of course, we need a bigger trials there, but these are, this is a big difference in signature there. You can see that it looks very, very promising there. There is a few more drugs that are generic that seems to have also an impact on decreasing outpatient risk. Most of the drugs we've talked so far has been for people in the hospital setting for the severe case. What about most of us? 80% of the population may have mild to moderate. So this is great news. This is a SSRI, which is a type of drugs there that can decrease by 85% the chance of people that, that will require hospitalization. So fluvoxamine is an antidepressant and OCD type of drug. If you take it within 7 days of symptoms, so again, in that period there where the body hasn't lost the, so fluvoxamine basically had a huge impact for people in a double-blind placebo control with 152 patients. So it's a good number there. And it shows very strong anti-inflammatory property against cytokine storm. Remember, we're trying to have that period between 7 to 10 days there to avoid the cytokine storm there. So this is very promising that we may be able to use these generic drugs on an outpatient basis. Again, vitamin D, every 2 weeks, I come up with more and more data. It is very clear-cut that vitamin D, there's so many data now from different research. This is a huge trial on around 192,000 patients there. And they saw a 96% inverse correlation of COVID positivity rate with vitamin D level. For anybody who knows about correlation, this is a really high number, 96%. You really get excited by that. And pretty much what he says is that the people who have deficiencies in vitamin D have a higher chance of getting severe case of COVID. So again, we talked about in the past, take a look at my video 2 weeks ago, please get vitamin D. And it was defined that if you do a blood test, if you're below 20 ng per milliliters, it's a deficiency. If you're above 30 to 40, that's kind of optimum level of protection there. And that's a blood test. There's this group of FLCC, which is a Frontline COVID Critical Care Alliances, which is a group of physicians in the US who are publishing protocols on how to take care of patients there. And if you remember, we talked about it 2 weeks ago with a math protocol. And they have now just come up with an update of that. And it's a little bit controversial. So they're recommending using a drug called Irvemectin. And Irvemectin is the drugs that you may be using topically for the lice. For, and, you know, if you're a mom, you may remember, for head lice. It's also used for intestinal infections. It's also used by the vet for basically infections, intestinal infections in animals there. Now, the FDA has issued a warning that this drug has not been approved for COVID. So what I'm going to say here is not something you should take in any form or shape. It's only recommended by physician in an ICU setting under their care. So what they've shown is that it inhibits that replication. You're in that period I've been saying from the time you get exposed to around day 10 there, you have that duplication of the virus. That's when you want to stop it. And that's really kind of the early action there. What they show is that within 48 hours in an infected cell culture, so it's in a petri dish there, they got rid of all the virus. They have shown some data there that it basically speed up the recovery of patients with mild to moderate diseases. It lowered the ICU and death rate there. So they feel very, very strongly there. And they're recommending to use this proactively there. But I want to reinforce that this is not a drug that has been approved for that use. It's off-label from an FDA perspective. So they, if you remember, we talk about the MASH plus protocol, which is using methylprednisone, which we know is what really works in the ICU setting there to stop the cytokine storm, using high-dose intravenous ascorbic acid, thiamine, which is vitamin B1, and heparin, which is to decrease the blood clot. That's the MASH plus protocol, which pretty much most hospitals are now using. The ivermectin, and this came to me by my friends in South America, has been really used outside the US. And if this is an interesting study has been published, where they show in Peru and in Brazil, they have done wide distribution of ivermectin. And they've found, if you remember, they had this massive uptake here that was really out of control there. And as soon as they start giving the ivermectin to the population, you can see they really got the infection under control there. It's unfortunately being really followed up in South America and other countries in Asia. And I really hope that, you know, the FDA and these clinical trials in the US to basically validate this. So the alliances, because they are physicians, they have the right to prescribe things off-label. They do recommend ivermectin for prophylaxis for people at high risk and that people may be immunocompromised. And please don't do this on your own. And then for people, of course, in early outpatient protocol after you have been symptomatic and have been maybe diagnosed positive there. So they are pushing this, but again, as I said, it is controversial. So I want to wish you again a happy Thanksgiving. Please stay safe, wear your mask, enjoy your friends, and I'll see you in 2 weeks. Thank you for coming.